Hello, I'm Christopher Wallis. I'm from the USDA ARS in Parlier, California. The main focus of my work is biochemistry of potato tubers, which sounds very complicated and complex. But the main reason why we're looking at how the potato plants respond to Liberibacter by changes in their physiology is it could lead to new discoveries and new ways to control this disease, especially from the breeding, potato breeding. In particular, I've teamed up with Charlie Rush's group at Texas AgriLife Research. They shipped me potato tubers to analyze for different chemicals that are involved in the development of zebra chip symptoms. In particularly, these compounds called phenolics, which are generally thought of as antibiotic compounds, also sugars and amino acids. And phenolics are important in the zebra chip because the freshly cut potatoes turn brown, and they turn brown because the phenolic compounds are oxidized by this enzyme. And much like when you bite into an apple and the apple turns brown, that's the same kind of process that's happening in these tubers. And what we have seen in the past is the phenolic levels are going up in the potatoes that have Liberibacter in them. We also look at the sugars and amino acids because sugars and amino acids react with each other when they're fried to form that wonderful zebra chip symptom. And we also found that when Liberibacter affects tubers, you get increases in sugars and some amino acids. We built upon our research from last year, looking at these things initially, to expand to 3 different cultivars, our continued work with 1867, as well as we're looking at red and russet potatoes. And what we found is, among other surprising things, is that red potatoes have the highest fresh zebra chip symptoms than the other 2 cultivars. And we also saw these consistent increases in these tubers of the phenolics, the amino acids and the sugars, when they become affected. With the greatest increase in all 3 cultivars between 3 and 4 weeks infection duration. So 3 to 4 weeks before they were harvested, there's a change in the amount of zebra chip you see at the end of the season. What we can do now is we can feed this information to breeding efforts. One good example is that with genetically modified grapevines, a similar pathogen called Wallis fastidiosa that causes Pierce's disease, they found a way to knock out phenolic production by modifying the grapevines. And when they do that, the grapevines do not develop Pierce's disease, even when they are definitely positive for the pathogens Wallis fastidiosa. So therefore, we could possibly use GM to knock out the genes that form phenolics in potatoes, and that would also probably reduce or possibly eliminate the development of zebra chip disease. And that's just one example. There's other efforts looking at different ways to limit these host responses to the bacteria that ultimately directly relate to zebra chip symptom development. And the key to stopping zebra chip in this case is not so much stopping the pathogen or the psyllid, but to prevent the host from developing these symptoms. So the potatoes are still marketable and still usable and good for the industry. And these are probably going to be very stable forms of resistance that we can develop that might apply for even more than just zebra chip disease. And so I hope to continue these efforts next year and keep building upon what I've done and possibly move towards some of these solutions.